Welcome, paramedic students, to the first lecture of Human Systems, Module 1, Lesson 1. This is the introduction to the human body covering Chapter 1 and Chapter 2, the chemistry of life, in your textbook from Patton and Thibodeau. You can follow me now and after your courses are done as EMS Guru on Instagram, Twitter. Also find me on SoundCloud as I upload some podcasts, and our YouTube channel, the EMS Chief at gmail.com. Welcome back to the Chemistry of Life lecture. We're going to get right to it. So we start off with the chemistry of life, starting with, as most of our discussions, at least the way I like to present them, is by starting with the zoomed in version and then zooming back out, meaning we're going to start with the smallest and most basic building blocks of a system, and then we're going to zoom back out of that system. So with chemistry, we start off with basically identifying the things that occupy space. Matter is anything that occupies space and has mass, and the molecules and elements that we're going to describe basically have those properties with subatomic particles. Molecules are going to be particles of matter that are basically made up of atoms. Atoms are the smallest unit of matter. So what we're showing here is protons, electrons, and neutrons as our list of the subunits of atoms, the basic subunit of matter. Well, let's identify some components or characteristics of each of these things. The proton, basically within its name, pro, is positively charged. The electron is the opposite of the proton, so its electrical charge is negative. And each proton has one charge, and each electron has one charge associated with it. Neutrons are neutral. So these things all come together to make up the atom. The nucleus of the atom is going to have protons and neutrons in equal numbers. If we have 4 protons, we would have 4 neutrons. And in an atom that is not highly reactive, you would have an equal number of electrons as well. But that can differ, and we'll talk about the cases in which it does. Here's our periodic table of elements. So I've given you a really brief introduction, but that's all we need to get started on our periodic table of elements. So the periodic table of elements is going to describe for us a set of periods and groups that put together different elements. The elements are basically the atoms themselves, so that we can organize them and describe properties that are similar among them. So along the periodic table of elements, we have groups along this top portion, and groups are basically going to be the columns. Then along the side, we can list that we have periods, hence the name periodic table of elements. So periods are going to be rows. So when we look at these things, they're all basically going to have some commonalities or trends among them. When we look at the trends of the periodic table in the group section, we can identify basically through numbering and organization different types of elements that have similar properties. So within this region here, number 1, we actually have a set of elements that are considered alkali metals. Group number 2 are alkaline earth metals. So you can see towards this side is metallic. And then we've got noble gases over here. In the center, sections 3 through 12, we have our transitions metals. And over here we have some common terms as well, but they're not used very much in emergency medicine. So we'll identify noble gas, and then we'll identify group number 17, which is basically going to be halogens. So when we're looking at this is going to be compositions that are closer to gas forms, and towards this side, solid metallic forms will be the representation. different groups have a tendency that along the group, so for a group it would be going downward, along a group we have increasing levels of organization and increasing levels of electronegativity. Periods also show the same properties, in which as a period goes on from left to right, the period also has increasing levels of electronegativity and organization. So really we're talking about more energy within these atoms. When we're talking about the atomic structure, we can also talk about how electrons are organized and how electrons interact. And this is important. So we have a nucleus for our atom. And let's say that we're going to deal with our carbon atom. Carbon has a set of 6 protons and 6 neutrons, and it should have 6 electrons as well. So when we look at the electrons, so in the nucleus we have protons and neutrons, but here outside of the nucleus, orbiting within these shells, essentially the electron shells, lives the electrons. So the shells are organized from the nucleus outward. And as we get further and further and further away, the energy levels of these shells changes. So the closest electrons, the first shell, so we have 2 of carbon's electrons here, they orbit in this first shell. The first shell can only take 2 electrons before it's full or closed. If it has 2 electrons, it won't interact with other electrons in this shell. So any further electrons, in this case carbon has 6, any further electrons would have to go in further shells. So we go another shell out. And in this case, we need to have 4 electrons in the outer shell. Those will all actually add up to 6 electrons, 2 in the inner and 4 on the outer shell. And those different shells have energies associated with them. Now I picked carbon because most of what we do in emergency medicine is going to be organic based, and carbon is the base of our organic chemistry. But if we go further and further through the periodic table, we would find that there's essentially 7 shells and then another number of subshells within them. The 7 shells are kind of related to the periodic table because there are essentially close to 7 periods. So when we're looking at this, and this is not necessarily something that you have to memorize for emergency medicine, but it helps explain. When we're looking at these shells, they're identified as K, L, M, N, and O as we go further away from the nucleus, right? Here's our nucleus. As we go further away from the nucleus, we have more and more shells. And as we get further away, more electrons can be held in those shells. We generally in emergency medicine will stick with the second shell level, or the L shell. The L electron shell can hold up to 8 electrons, and we generally would call that an octet of electrons. Now, an interesting thing about these shells is that they really want to be closed, and that means that when the shell has less than, for our cases in shell number 2, if it has less than 8 electrons, it's seeking out bonds to fill those other electrons. In our case, with carbon, we have 6 electrons total, but only 4 are in this second shell. And we know that the second shell likes to have 8, or our octet of electrons. So that means that we have 4 electrons in that shell. We could take 4 more electrons to make this carbon atom satisfied. The valence shells are where the strong bonds actually are made. So valence shells need to be separated and differentiated from just our electron shells. They are electron shells, but they're a specific type. They're the outermost electron shell. So in our case with carbon, having our first lining covered and full, and our second shell open to receive 4 more electrons, we would consider this the valence shell. So the valence shell is also going to refer to a property of chemicals, the valence property. And the valence property aptly named, really talks about how readily a element or atom will bond with other elements. And that's really based on how many open spaces for electrons are within the valence shell of that atom. So the valence shells like to be closed, as I mentioned before. If they're closed, then the element is inert and not easily reactive. But when they're not actually full, they seek to get more electrons from other means. All right, continuing on the description of atoms and the periodic table of the elements, we're going to look at one tile or one atom within the periodic table. So we've talked a little bit about carbon, and this is in fact what we're identifying. So when you look at the periodic table of the elements and you look at a specific element, the letter that's center and probably bold inside that tile is the abbreviation for the element. And in this case, as you probably know, it's carbon. Now, we've also got some other useful information, and periodic tables can get quite more in depth than just this on a tile, but this is the basic format we need for EMS. This is going to be the atomic number. The atomic number represents basically the number of protons and the number of neutrons within the atom. And that should probably equal the number of electrons. This number here is the AMU, the atomic mass unit. The atomic mass unit is basically going to be an average of the weight or mass of all, in this case, carbon isotopes. So isotopes are a unique term, and in New Mexico, they're the name of our baseball team. But isotopes are going to refer to the same element, but an element that has a different number of neutrons and protons than the original structure. And these isotopes can be very useful in medicine and in things like dating of specimens and so forth, but we won't use them in EMS. We'll just highlight what that information is. So now we can identify, just with this information, how many electrons should be available. And remember, first we'll fill the first shell with 2, and then the next shell can hold up to 8. And if we have less than 8, then we'll be filling the first shell and leaving the second shell partially open. Here's a list of some common elements that are useful in EMS. The name, abbreviation, the atomic number, and the number of valence electrons that are in each element's valence shells. This is not an all-inclusive list, but these are the most common chemicals that are used to describe either pharmacology or physiologic processes in the body that we have an impact on in EMS. Now let's talk about ions. Now ions are not the same as isotopes. Ions are describing a charge associated with each different chemical. Some chemicals can lose or gain their electrons pretty easily. And so an ion is an element that's not neutral. And that's what we mean by this has a non-zero charge. If an element has a zero charge, then it's just an element. But in this case, we're talking about something that has a charge. So number one, we have positive and negative charges that can occur. To memorize the ions, you want to memorize that the cation has a big old plus in the middle of it, right? Cation. That's a big old plus, so that's positive. When we talk about anion really kind of uses the term a or an, and that can mean, in terminology, without. So if it's without a positive charge, then it's a negative charge. So how do we get to these things? Well, we can have chemical and physical reactions that occur that cause an element to become an ion. Now, an ion is going to be created by a change in electrons. So in each case, we can identify what happened. So let's look at this first. The cation has a positive charge. A cation has a positive charge because not anything to do with its nucleus, but it lost an electron. If it lost an electron, then that negative charge, that one negative charge that the electron had, is not opposed. And so now only the positive charge of one of the unimpaired protons in that structure are going to relay the charge. And so it's positive because it lost an electron, but it kept that positive proton that it should be paired with. The negative ions or anions get their name because they gained more electrons than they have protons. So if, say, carbon were to become an ion, and it wouldn't, but if carbon were to become an ion, and that's what our example is, if it has 6 protons and 6 electrons, for it to become an anion, it would have to have 7 electrons and 6 protons. And that 7th electron would be unopposed with a positive charge from a proton because there's more electrons than protons, so it would be a net negative charge. An important note of how we identify, let's look at one of the ions we commonly work with, sodium. So sodium has a positive charge of 1. And so the way that we identify this is by writing it as a 1 and a plus. You can also, if it's only one charge, write sodium with 1 positive sign. Some others, like calcium, have 2 positive charges, and sometimes they're written as calcium with 2 pluses. Either way, what we're identifying here is that calcium is short, lost 2 electrons, and sodium lost 1 electron, giving rise to the idea that its unopposed positive charge inside the nucleus is showing as 1 positive charge overall. The characteristics of ions, and we've listed here a couple common ions and their charges within emergency medicine. The ionic elements generally are not attracted to one another, so they repel each other if they have the same charge. So positive and positive won't happen, but they are attracted to opposites. Just like the saying goes, opposites do attract. That means that when we have ions coming together to create stability because they're missing an electron, they want to find an electron, we can build elements and molecules like sodium chloride. And let's see what happened here. We can see that chloride has 1 negative charge and sodium has 1 positive. So that means the chlorine element has 1 more electron than sodium does. Sodium has 1 less electron. So if we have a positive 1 charge and a negative 1 charge, and we bind these things together with an ionic bond, we end up with no charge whatsoever, and this ion has now become a salt. So salts are ions paired together. Notice there's no charge written here because sodium's 1 positive and chlorine's 1 negative together result in no charge. When we put things together like we just did with our ion, we build up from elements. Elements, remember, were only one kind of atom, and generally they're going to be very simple atomic forms. Now, if we get complex and we add a couple of different atoms together, then we can end up with something like a molecule or a compound. A compound generally has more than one type of atom associated with it. So a really good example of a compound might be H2O. A molecule could just be an elemental substance of oxygen. So let's look at how these things pair together. Chemical bonds are going to arise from different properties within these elements. And we have really 3 types of bonds, but we talk about 2 major types. We have the ionic bond and the covalent bond. So right off the bat, you should get the idea that this is covalence shell. So this means that the valence shells are connected together when we have this bond. The ionic bond is based on that positive and negative charge, resulting in something like a salt. And then, as mentioned, we have one more, and that's basically going to be the hydrogen bond. That's a weak bond that's somewhere along the idea of ionic bonds in which hydrogens close together can attract each other depending on certain principles. We'll see that a little bit later. Now, when we talk about chemical bondings, we can end up with electrons being switched and shared between different atoms, or we can have just loose associations. Let's start with the loose associations. So remember that the positive ion is called the cation, and the negative anion is called the anion. So when these things are coming together, they're generally being attracted to each other by their equivalent charge. When we form these ionic bonds, they're basically going to create a salt, as I mentioned, and if that salt is going to be dispersed in water, then we've created an electrolyte. So that's a really big EMS term, electrolyte. In emergency medicine, we see electrolytes being discussed in the fluids that patients are receiving, in imbalances that are the result of maybe dehydration, and the result of that imbalance of electrolytes can be severe, including abnormal cardiac rhythms and some lesser severe forms like cramping and otherwise. So when we have an electrolyte, we're really describing something that when placed in water is going to easily break apart. So remember that we had sodium chloride, right? Sodium chloride is table salt. Table salt. So it's the same thing that's found in the ocean, in salt water, and it easily pairs together. But when this is inserted into a glass of water, sodium is going to dissociate from the chloride and float around freely. This is important because we are primarily made of H2O. And so all the chemicals we talk about in our body are going to be put into a solution that has water as a prime component of that solution. So electrolytes, when given, dissociate. That means that if we need sodium and we give somebody sodium chloride, also known as normal saline, when we give them that drug, truly what it is, that IV solution, once it enters the bloodstream, sodium and chloride are separated and become available for use as independent elements. Here's a picture of some bonding that's occurred in ionic bonding. So here's sodium chloride, and this is basically the way that sodium chloride naturally forms a cube-like structure and creates salt crystals, just like you might find laying around on your table. If we zoomed in, this is what happens, and notice that we've got chloride is a pretty big element. It's got 17 as its atomic number, meaning that there's 17 protons on the inside of that nucleus. So there's a lot of energy shells in which the outer energy shell is indicated here. Now when we have sodium's a little bit less complex. We have essentially only the valence shell being the second shell outward from the nucleus of sodium. These are not physically connected in the diagram. The bond between the two is virtual. There's just a loose attraction. Covalent bonds are different. Covalent bonds are going to include bonds that have very strong properties. They're hard to create and hard to break up. In fact, they generally require energy. So what ends up happening here is not just a loose association, but we end up having, if we have a valence shell of this atom, the valence shell of the bonded atom would actually touch and form a bond sharing their electrons. Now this is helpful because if this atom, we'll say atom A and atom B, if atom A needed 4 electrons and atom B has 4 electrons, then the 4 electrons that atom B gives can be added to the atom A electrons, and maybe that closes or satisfies the valence shell, especially if we're talking about that second valence shell. So let's kind of look at how this comes together. Let's start with a very simple molecule, the oxygen molecule. So the oxygen molecule, as you know, is basically 2 oxygen molecules together, and it's generally in a gaseous form. So let's draw that out as it would be in showing its bonds. So I'm just going to show the nucleus of each of these without showing the protons and electrons. But first, we probably should identify that when we look at oxygen, oxygen's atomic number is 8. And so when we're looking at oxygen, it's got 8 protons, 8 neutrons, and should have 8 electrons. And let's imagine how those are separated. The first level is going to be operated with 2 electrons, and the second level will have 6 electrons, meaning that we could take 2 electrons on and complete our shell. Remember, we want 8. So let's draw our first shell. We got electrons here, right? And circular. First shell, happy. Remember, we have 6 electrons in oxygen. So we've got, in the valence shell rather. So what's going to happen is we want to satisfy this. So we're going to share 2 electrons from this oxygen and then share 2 electrons from the other oxygen. That gives us 4, and those 4 count for both. So 4 for this oxygen and 4 for this oxygen, even though they're sharing 2. Now, we still have a few more that we have to account for. We shared 2 of the 6 available electrons here. So we still have 4 electrons in this shell that aren't used in bonding. But now we have 4, 5, and 6, 7, and 8, and we're happy. On this side, we have a similar setup. The other electrons, totaling 8, makes this molecule very happy. So if we use a Lewis structure to identify this, we would show basically oxygen and oxygen, and they formed 2 electrons bonding together. That means that we have a double covalent bond, and that's shown in the Lewis diagram with 2 lines. So we don't have any other electrons available to pair with something unless this breaks apart. Now, because we're sharing electrons, this is really strong. So now we're going to introduce something called the easy come, easy go proposition, or as we'll say it's the Schwann theory. Schwann theory. How about that? So the Schwann theory is that if something is easy come in the human body, in emergency medicine, otherwise, it's going to be easily gone. So let's think about this. If we're using this to describe ions, then ionic bonds form very easily, but they don't last very long. They're not very strong. So something that forms easily goes away quickly. This, on the other hand, is very strong. This, oh, and I should separate this. That is not an extra bond. We only have 2 bonds here. But this is very strong. So it's not easily put together, and it's not easily broken. These are the bonds that we want for really strong chemicals that are going to be durable to do the work of the cells for our life. Let's look at another example. Now, we've looked at carbon and we've looked at oxygen, so now why don't we bind these things together to create carbon dioxide? So what we're going to create here is basically a carbon with 2 oxygens attached to it. So this will be our carbon nucleus. We'll say carbon nucleus. And this will be our oxygen nucleus and oxygen nucleus. All right, so remember that C is going to have 6 protons, neutrons, electrons, and oxygen is going to have 8 protons, neutrons, and electrons. So we'll organize those accordingly. If we're looking at the shells, the first orbital shell of each of these will not be shared. Because remember, since we have more than 2, the first 2 electrons are going to be used in those orbitals. So for each of these, we have an electron, we have an electron, 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 electron. So remember, that's in this first shell. Now we're moving on to the second shell. And the second shell is going to be the shell that we'll share. So these are all valence shells in the case of carbon dioxide. With carbon binding to oxygen, we're actually going to have double bonds. So we've got 2 from carbon and 2 from oxygen on one side, 2 from carbon, 2 from oxygen on the other side. Now, as far as carbon is considered, that's 4 and 4, so that's 8. That makes carbon happy. Now we still have to do the same for oxygen. So oxygen has 4 here, and then just as we did when we paired the oxygen with oxygen, we have 2 pairs of electrons that are not used in binding. Now we have 1, 2, 3, 4, 5, 6, 7, 8. That means that the oxygen is happy and the oxygen is happy. And if we draw this in a Lewis structure, we would draw something like C with a double bond to oxygen and oxygen with a double bond to carbon. And then we've got those other electrons that we have to identify. and we're set. Carbon dioxide is likely not going to be available to pair with other things because its valence shells are closed. All right, now let's continue on. I know we're doing kind of a lot of the chemistry here, but these are all important structures related to how the cells work later on. So glucose is a little bit more complex. So I'm going to draw glucose in kind of a few different ways. So number one, we have to identify glucose's chemical structure. So the chemical formula for glucose is 6 carbons, 12 hydrogens, and 6 oxygens. So we have to account for this when we're putting together our diagram. Let's start with a Lewis diagram. First, we want to draw basically 6 carbons down a chain. Each of these carbons have a single bond to each other. So again, this is our Lewis diagram. Now, one end of this glucose is going to have an oxygen with a double bond and a hydrogen with a single bond. And as we go through, we're going to see a collection of hydrogen and this new thing that we introduced, OH, which equals, let's put it here, OH has a negative charge of 1 and is a hydroxide ion because it's got a charge. That pairs with the single positive charge of hydrogen ion. Carrying on, so we've got that collection of hydroxides on one side and we have hydrogen on the other. For time's sake, I've created basically the hexagonal shape of glucose as it really would exist. And so in this three-dimensional structure in the hexagonal shape, we end up with a structure that can be further simplified to just be drawn as this and mean the same thing. And so what we've created here is one glucose molecule. And if we created one glucose, we probably created something like a mono, meaning one, saccharide, referring to sugar. So this is important because of what we're going to talk about in a few slides, but we're still on the idea of covalent bonds. So we can see here that we've created essentially covalent bonds to create the structure of glucose. And as you know, glucose is a carbohydrate, and in some forms, carbohydrates are very, very difficult for us to break down as humans, and we actually need to have some help. So covalent bonds are going to create very strong bonds between two atoms. They may be similar and they may be different by bonding electrons that are shared in their overlapping valence shells. Here's a picture of those very loose bonds created by hydrogen bonds. So here we have water floating around and water molecules floating around in solution. So we can assume that this is probably some form of water in fluid. And when these oxygen and hydrogen molecules are floating around and interacting with each other, they have some loose bonds that occur when they pass by. Now, this is actually not the only place that we'll talk about it. At the end of this slide set, we'll actually talk about how hydrogen bonds are important in DNA structure, but we'll save that for later. So we've up to this point been talking about organic chemistry because organic chemistry generally involves the use of carbon bonds with other types of elements, very often carbon and hydrogen. If we're talking about an inorganic chemical basically are trace elements and other things that are not associated with carbon. So water is inorganic. Some acids that aren't created by the body, bases and salts, can be inorganic. And then we can even look at things like minerals, which are just another way of describing elements. Inorganic chemistry accounts for water as being one of the most abundant things in our body. Oxygen, carbon, hydrogen, and nitrogen are actually the most abundant elements in the human body. And you can imagine if we've got carbon, hydrogen, oxygen, and then nitrogen we haven't talked a lot about, we have the building blocks of basic structures that build a cell from your knowledge of taking biology and things like that. Some of our base elements are actually trace elements, but although they're trace elements, they are extremely important in the normal process of human physiology. As stated, we are mostly water. And so when we talk about water in the human body as EMTs and paramedics, we can very easily account for how much water the person should have in their body because we can use some standards and constants. So in the healthy human body, the male and the female have different compositions of water. In the male, they have 60% of their body weight accounted for as water, and in the female, about half. So when we talk about water, this is what all of the chemicals that are in our body are going to be dissolved in. Remember, electrolytes are going to be dissociated in water. If we need to figure out how much water somebody needs to have in their body, there's a very easy formula. The first thing to identify is that 1 liter of water weighs 1.01 kilograms. So if you have somebody's total body weight in kilograms, then that equals their total body water in liters. So for time's sake, I won't actually calculate my body water content, but I implore you to pause the video and take the time to calculate how many liters of water should be in your body based on your weight. You'll probably be pretty astonished at just how much water that is. But remember, in any case, that 1 kilogram of water is equivalent to 1 liter of water, and 1 liter of water weighs 1 kilogram. Remember that electrolytes, as mentioned a few times, are going to be the salts of ions that dissociate when put into water. Water is the solvent, the liquid in which the solutes, or things like sodium or glucose, are going to be surrounded by when put into the body. So remember, cations, positive charge, anions, negative charge, and that's important because any salt will be dissociated when entering water in the human body. So water is important because water reactions are something that are the building blocks of the basic molecules and structures of our body. So we can't talk about all the different types of molecules in our body without first talking about what water does to it. So water is going to be important in the process of building and tearing down really complex molecules and structures. And so dehydration synthesis and hydrolysis are basically opposites. So memorize them as opposites, basically opposites in that process of building and tearing down big chemicals. ATP requires water and its involvement, and chemical equations are generally going to highlight how things occur as a chemical transforms from one or multiple chemicals to a new set of chemicals with new compositions. So why don't we look at actually how this happens? So the first thing we're going to talk about here is water-based chemistry is going to be the use of hydrolysis on one hand. Remember, they're opposites. And dehydration synthesis on the other. So hydrolysis, let's break the terms down just like any new language. Hydro, H2O. Lysis, generally when we talk about a cell lysing, it generally breaks apart, sometimes explodes. So it breaks apart. Lysis equals breaking apart. So what we want to get from that is water is involved in breaking apart the structure. Now the opposite, again, versus, is dehydration synthesis. So dehydration, we remove H2O, and synthesis means basically we build. So we build chemicals by removing H2O in dehydration synthesis. So let's look at that. So remember our glucose structure, right? We got our hexagonal glucose structure. This is 1, so this is a monosaccharide. The monosaccharide by itself is useful inside of a cell, but we have to store sugar based on a feedback loop. Do you remember which feedback loop I mentioned? Good, it's a negative feedback loop. So if we have to store sugar for later, let's say that we ate food, we took in the sugar. We have two pathways. We can use the sugar or we can store it. When we store the sugar, we're going to store it as a more complex form of the carbohydrate. So what we're going to do is add our monosaccharides together. And we're going to create things like, if we have 2 of them, we have a disaccharide. If we have more than 2, then we have a polysaccharide. And polysaccharides are going to be long chains of these monosaccharides linked together. So we built that. Remember, we had to build this. So if we built this thing, then we used primarily dehydration synthesis. Remember, building. So how does that work? So we take a glucose structure and we zoom in a little bit. So let's zoom into this glucose. So remember that we have a OH and we can also have an H group, right? And here's the rest of our monosaccharide just for purposes. So right now they're individual. They both have these structures. So we take this OH and this H, and you might be able to see what's coming here. And what we're going to do is dehydrate by removing the hydroxide ion and the hydrogen ion from the glucose. The hydroxide has 1 negative charge and the hydrogen has 1 positive. And the equation that we then get is going to be, we have 2 hydrogens and 1 oxygen. So we've created water. And in that process, we got rid of these hydrogens and we created a bond. So this is dehydration synthesis, right? We removed water and the water now is free floating. And in doing so, we removed the water and bound these monosaccharides together to then create a polysaccharide. Now let's say that we ate a while ago and some time has passed, right? Time has passed on the clock and we ate a while ago, and now we need to break down some sugar. It's in the polysaccharide form right now, maybe in our liver or something like that. And we need to remove one of these so that we can use it in a cell. So let's say that we want to break this one down right here. So remember that we removed water in order to bind them together. Now with the term hydrolysis, we're going to add water to break them apart. So H2O is used and added into this. And when they break apart, we get individual monosaccharide over here. And let's say we still have our polysaccharide chain over here. And remember, since we broke it apart, we ended up with an OH and we ended up with an H on this group. And now they're free floating. The monosaccharide is available for you to eat and replenish your sugar, again with the Cyclops guy. So this is hydrolysis. We added water and we ended up splitting a complex sugar into a very simple monosaccharide so that it could be digested. That's the essence of what happens during the process of water-based chemistry when we're talking about building and breaking down complex structures. Now, another component or characteristic involved in water-based chemistry is the idea that we have acids and bases within the solution. So remember, the solution for us is water, but this is just an example of different types of acid and bases, not necessarily all from the body. So let's kind of put this in perspective. The pH is going to stand for the potential of hydrogen. And just, if you're wondering, that's a D. All right, potential of hydrogen. So when we say potential of hydrogen, this is a tricky thing. The hydrogen ion has a +1 charge, right? And the hydrogen ion is floating around freely. And hydrogen ions in abundance, or the concentration, that's what these brackets here mean, the concentration of hydrogen ions is going to tell us a little bit about the acidity. So if we have a lot of hydrogen ions floating around, then we are acidic. But the relationship is a little bit tricky. This is an inverse relationship. Put a star here. inverse relation. So that means that the numbers on the scale are inversely or oppositely related to the value of hydrogen ions. That means that a lot of hydrogen ions is 0 on our scale. So there's another piece to put in perspective here though. If we have hydrogen ions in a solution in the body, we also have hydroxide ions. So what this is saying is that the concentration as we get closer to 0 of hydrogen ions is high, but at the same time, we also have a low concentration of hydroxide ions. And then over here, if we say we have low hydrogen ions, we have more hydroxide ions. And so that's the proportionate relation. And that's important because if we have more hydrogen ions than we have hydroxide, then we have an acidic concentration. But if the pH value is red, the higher the pH value, high pH equals low hydrogen ion equals low acid. That's the relationship. So in your brain, spend some time trying to get that together. Our scale is from 0 to 14, and in the middle is 7. Now 7 is perfectly in the middle of 0 and 14, and that's important because at the point of 7, given that what we're really looking at is the concentration of hydrogen ions and the concentration of hydroxide ions, when we're at a point of 7, there's an equal number of hydrogen ions for an equal number of hydroxide ions. And that produces, for every one hydrogen ion, if we have one hydroxide, that creates water. And pure water is neutral. So that's essentially why we have this picture of an inverse relationship, because we're talking about not just the hydrogen ion, but the hydroxide ion. So in your mind, be able to equate that a 7 pH value is neutral because neutral in acid and neutral in base, because we have an equal number of hydrogen ions to hydroxide ions, and all of them paired together because they're ions to create water. And water, as you notice, doesn't have a charge. It's neutral. So let's look at some of the things on this list. It might surprise you to know that vaginal secretions are very acidic. It might also surprise you to know that along this line is where household bleach is found. Household bleach is a base, very damaging base, but we find in emergency medicine later when we talk about the skin and burns that bases are actually a little bit more severe in the burn they produce in soft tissue than actually acids would be. So be able to relate this idea that if I give you a number, say a number 8, you can tell me that that is not in fact an 8, meaning it's high in hydrogen ions, but it's high in hydroxide ions and low in hydrogen ions. So the closer to 0, the more acidic. The further away from 0, the more basic. We're going to talk a lot more in depth on the pH scale and how that's influenced by the way our body works and the buffer systems associated with it. But for right now, that's the gist of it. So here it's kind of restated in a different way, but again, remember it's hydrogen and hydroxide. Also on this slide, it's important to identify that when we're talking about bases in medicine, we usually use alkali or alkaline instead of the term base, but they are interchangeable and you should be able to use both. The buffer systems are kind of important. So the buffer systems in our body are really kind of ranked in order of increasing effectiveness, but they follow the easy come, easy go rule. So our buffer systems are made up of the blood buffer, the respiratory, and the renal buffer system. So again, increasing ability to deal with acid as we go more towards renal. Blood is based on hydroxide ions floating around in the blood. So easy come, easy go. First to act, first to go away. So that means that if our blood buffer system is working instantaneously within seconds, that hydrogen ions start to accumulate in larger numbers in blood, the available hydroxide will create water when binding with those hydrogen ions, and you'll breathe or urinate that water off. Remember, the water was neutral though, so it helps to mitigate the acidity of blood when we have a lot of hydrogen ions. But this doesn't last for very long. This will last for a few minutes at most. If we're still creating hydrogen ions, then we'll move to the number 2 buffer system, and that's the respiratory buffer system. And you blow off CO2 as a means of mitigating the acid concentration. This happens minutes after the increase in hydrogen ions, after blood buffer system's been used up, but can last for a period of sometimes even into hours. The faster you breathe, the more CO2 you blow off, and the more CO2 you blow off, the more hydrogen ion and acid you blow off. Let's say that that doesn't do the trick and you're still accumulating acid. Your renal system is kind of the final prep. The renal system takes hours to kick in, but when it kicks in, it's very effective at peeing off hydrogen ions by extracting it from your blood and entering it into the kidneys and eventually voiding it in urine. Important to review these, especially as an EMT basic moving on to another level, because the medications that we give and the fluids that we give do impact the buffer system's ability to work. All right, now to get to our final section of slides, we're going to talk a little bit about the different types of chemicals and the classifications that we need to move on to biology. When we talk about the cell in organic chemistry, we can break down 4 different categories that most chemicals fall into. So we have carbohydrates, right? These are sugars. They use the monosaccharide, which we talked about earlier. The lipids equals fats. Proteins, well, you know what proteins are. And the nucleic acids found in DNA and RNA. So an important note, let's put a note here. What I want you to know for all of these is 1, the class, 2, basic units in that class, and 3, give an example of these chemicals inside the body and what they do. If you can do this, you're very well prepared for any exams that you'll have to take in class and for licensing exams as well. So let's go through each of these 4 categories and identify the structures within them. So organic chemistry really means carbon and some other chemical. When we're talking about carbohydrates, carbon, hydrate, water, it quite literally means carbon and water mixed together. And we saw that because the ring structure showed that we have hydroxide and hydrogen ions linked to carbon and some oxygen as well. Monosaccharide is 1. That's our simplest sugar, and that's what our cells need to do work. So normal dextrose fits this because it doesn't have to be broken down any further. This is important when a person is having a diabetic emergency. Disaccharides are a little bit more complex, but not by much. They have to be broken down eventually to the monosaccharide. Here's a question for you though. If we're breaking down a larger structure of, in this case, 2 to 1 saccharide, what chemical process is it? Well, remember, to break down, we've got to add water, and that breaks down the structure, and so this would be hydrolysis. Forgive my writing, it's getting late. So polysaccharides are going to be more complex than our disaccharides, and the polysaccharides are going to be very complex carbohydrates. In the human body, we store glycogen as our source of polysaccharides. So when we're using dehydration synthesis to build monosaccharides into polysaccharides in our body, we are building glycogen stores. In the world, it's not stretch, it would be starch. Apologize for that misspelling, but that's the plant world version of polysaccharides. Here's a picture of the different types of carbohydrates and representations. Now the next thing we talk about would be the lipids. Lipids are going to describe fats and oils in the human body, and we have a few major classes. Triglycerides are going to be important types of lipids that are going to be sources of energy. They're going to have 3 fatty acids, so that's where the tri comes from, and the glyceride is the base unit. So here's this little piece of our puzzle, the second piece to all of these organic chemical classifications. Glycerol is the base unit for lipids. Please do not confuse glycerol with glycogen or glucose. Those are carbohydrates. So glycerol is the base unit. Now, phospholipids include a phospho or phosphate group. And the phosphorus group is going to add to the properties of this lipid. Lipids that are phospholipids have a head and a tail, and the head and tail both attract and repel water. This is an important component of cell biology. In fact, without this thing occurring, it's hard to imagine that life would have occurred on Earth. Now, cholesterol is going to be one of the least favorite types of acids for most people, but realize that cholesterols are involved in the creation of steroids. Steroids are hormones. They're important in chemical messaging. So we have to have cholesterol in our body. They're also going to be very functional and structural. We can find cholesterol buried in some of our plasma membranes, for example, and used in processes that will help communicate from the outside of a cell to the inside of a cell. Here's a picture of phospholipids, and this is basically the plasma membrane of a cell. The plasma membrane exists because the phospholipids are going to organize themselves to meet their tendency. The head likes water and the tail does not. So when placed in water, these are going to all organize so that the tails face each other and the heads do not face the inside of the plasma membrane. They face the water. So this creates a tight bond between these that prevents water from getting into the space. In fact, if water is injected in here, these will flip and destroy the plasma membrane. Proteins are the third category of structures, and in proteins we identify a couple different characteristics. Proteins are going to be made up of amino acids as their base unit, amino acids. We don't have to memorize all the amino acids nor all the nucleic acids for our level of EMS, but it is important to understand what an amino acid is. So an amino acid obviously has an acid quality associated with it, but when we look at proteins that are made up of amino acids, they can end up as structural proteins, or we can also have functional proteins like enzymes. Let's look at this. This is a picture showing the organization of complexity when folding proteins. The first level is the primary folding level. So the primary structure is basically a sequence of amino acids making up an amino acid chain. There isn't any folding involved. The next level is the secondary level. The secondary level is when we take this same chain and we fold it into pleated sheets or twisted helices. Twisted helices can be important because the central canal of that helices can be used to transport things. The tertiary level of protein folding involves a protein structure that has multiple twists and folds added to it, creating a three-dimensional structure in space. Now, if we move on to the quaternary level, this is the most complex folding level of proteins. This is when we have more than one folded amino acid chain that's probably undergone up to tertiary folding bound together. And the best example of quaternary folding in the human body for emergency medical services purposes is going to be hemoglobin. Hemoglobin is a quaternarily folded chain that has an alpha and beta section to it. And these two three-dimensional structures together help create the binding sites for oxygen within hemoglobin. Please do memorize these levels of folding because the change in acidity of blood or other solutions can impact the folding of proteins. The functional proteins that were mentioned earlier are going to help do some pretty amazing things. Enzymes are generally going to help make some chemical process occur. It probably occurs already, but it helps some chemical process occur or react more efficiently, more quickly, or perhaps makes a chemical process occur that couldn't occur without the enzyme as a catalyst. The enzymes also can act as a type of lock and key. This is particularly important when we talk about insulin. And then also hormones, cell membrane channels, receptors, enzymes all have some type of protein associated with them. Proteins can actually be folded and attached to other structures. If we attach a protein to a carbohydrate, we get a glycoprotein. If we attach a lipid to a protein, we get a lipoprotein. These can be involved in the construction of the plasma membrane and for receptor sites in the human body. Finally, the last category associated with organic chemistry molecules is our set of nucleotides as the base building block for both DNA and RNA. DNA and RNA are very closely tied together, and each of them are going to have a phosphate unit, a sugar, and a nitrogen base. So when we describe this, one of the big differences between the two is actually the sugar. RNA is going to have a ribonucleic acid because it uses ribose as the base sugar. DNA uses deoxyribose as a component of the nucleic acids. We don't have to memorize how these things organize themselves together, but what we do want to highlight is that DNA is going to be the master code in a cell. It tells the cell how to reproduce. It tells the cell how to do the normal functions of a cell through day-to-day life for that cell. RNA is going to be the copy made to do work from DNA. DNA will stay within the nucleus, but RNA can translate that information. As it were, open the book of DNA, the book of life, read from the page on how to build a new plasma membrane, copy that page, close the DNA book so it doesn't get damaged, and then take that copy it made into the cell so that the working components of the cell, the organelles, can get their jobs done. DNA, excuse me, as highlighted, forms a double helix shape. And remember that as described earlier on, the double helix shape is held together using these loosely associated hydrogen bonds. And this is important because if you think about it, the helix shape is going to have to be unwound to be read. And so we have to peel apart the helices, and we're going to use some proteins to basically write down a copy of the DNA. And then we zip it back together. But we wouldn't want covalent bonds here because it would take a lot of energy and may result in more mutation and errors to separate the DNA if they had covalent bonds. But hydrogen bonds are pretty loosely associated, so they tear apart, or we should probably say unzip more easily without energy and without having to decrease the genetic code. RNA, as mentioned, is the working copy of the gene that's taken from DNA and then used by structures within the cell. Finally, we rest with ATP. Adenosine triphosphate is going to be the powerhouse of our body. It's essentially going to be the little packet of energy, just like this picture shows, that our body uses. And this is a very simplified process of metabolism and cellular respiration. When we're looking at this, we need a couple components. We need glucose from food, and we need oxygen. And in that process, we're going to create ATP, adenosine, with 3 phosphate groups attached to it. The phosphate groups are really the energy that's used because the phosphate group makes the thing do whatever work we're talking about. In this case, the bicep is going to contract, and in losing a phosphate group, it becomes adenosine diphosphate because 2 phosphate groups have high energy bonds associated with them. More on this a little bit later. We're going to go into depth on both metabolism and on cellular respiration and talk about how that yields ATP. And all through our lectures on the human body, we will explore how ATP influences virtually every body system that we need to get something out of or need to rectify in emergency medicine. So that's the last slide for us. I appreciate you guys spending your time with me. If you're moving on to the next lecture will be covering